Organ on chip system was the best system that I could ever imagine to fulfill my aim. Hi, I'm Puja Pandey. I'm from Hannover Medical School in Central Animal Laboratory, and here I'm working as a postdoctoral researcher. In my PhD work, I work with different animal alternatives in order to understand the epithelial cell regeneration in intestine. As an alternative method, I work with 2D cell culture, 3D intestinal organoid culture, along with organ on chip system, where I work with primary epithelial cell, endothelial cell, and stromal cell and co-culture them in the chip. And now, as a postdoctoral researcher, I want to introduce immune cells such as macrophages so that we can mimic the intestinal structure. The main research question that we want to answer is to find best in vitro model that can be used as alternative to animal testing. And the second one is we want to know if our established organ on chip system can mimic intestinal environment. And third important question that we want to answer is if our established system can mimic disease modeling. The main reason to choose organ on chip model for my research is to overcome the limitations that we have in traditional cell culture. Like for example, in 2D cell culture, it doesn't have 3D structure, but naturally we see our cells in our body they are in 3D structure. Likewise, we also work with 3D organoids, but 3D organoids are enclosed structure and they lack vascularization, they don't have waste removal system, and they don't have this dynamic process of continuous medium flow. That's why all these limitations, it's overcome by organ on chip system, where we have perfusion system that continuously helps medium flow, that gives nutrients to the cells in the chip, as well as with perfusion, the waste materials are also removed in the system. And also with organ on chip system, we can co-culture different cell types. And these features are absent in 2D cell culture and 3D organoids. So the main aim of my project is to establish the system that mimics intestine morphologically and functionally. At first, I worked with organoid model where I simulated them, but we have many demerits of organoids and there are limitations. That's why I worked on organ on a chip system. Here, for the first time, I established a murine gut on chip model where I seeded primary epithelial cell, stromal cell, and endothelial cells together, so that the arrangement of the cells in the chip mimics the arrangement of the cells in the colon. Here, I confirm the compatibility of these three cells together by doing immune staining, and here you can see staining for E-cadherin, that is for organoids, and COX-2 for stromal cell, and CD31 for endothelial cell. So from this we can say they are compatible with each other together. In the next step, what we wanted to do was to analyze the epithelial cell regeneration after infection and treatment on chip, and for that we infected the chip with E. coli Nissl, which is a probiotic bacteria, and from that we could see after 24 hours there was a slight damage in the cell layer formed. But if we see the results of qPCR along with damage, we can see that there is increase in proliferation and increase in R bonding. That signifies that there is regeneration starting already, even though there is damage. We had Krupp's formation when we used Kakotu cells. So in that we could see really nice Kakotu, and then I made transverse sections of them, measured the height of the crypts, and we also carried out experiments like stimulation or infection with E. coli Nissl and also pathogenic bacteria Citrobacter rodentium. I find quite interesting that even after the damage, we can see regeneration of the cells like instantly. All the inflammation as well as the recovery is happening at the same time, and I find that very interesting. So if we compare it to traditional culture methods such as 2D cell culture and organoids, it is quite complex. But if we are able to avoid formation of bubble or avoid contamination and avoid leakage and work properly with care, it is quite easy and it is quite user friendly. I feel the recent chip that we got from Dynamic 42 is quite easy to handle, and it is best to culture different cell types because it has porous membrane, and then we can culture different cell types on upper and lower chamber. So the main problem I faced was in the starting with formation of bubbles, because I was not that perfect in pipetting the medium or cells inside the chip, so I had this problem of bubble formation. But later on I excelled my pipetting skills, and then that problem was solved. And another problem I faced was I wanted to measure the tear value in my chip, but with the chip that I am using right now, it was not possible to measure the chip. But I recently found that it is possible, and I'm really excited to use it. And the next problem I faced was the leakage of the medium from the old perfusion setup, and for that we changed all the old setup, and it was because there was wear and tear in the connectors, and that's why we changed that, and then after that we solved that problem. Another problem that I faced was imaging the chip directly, and because of this plastic bottom chip, it was not possible to get beautiful pictures. And I am so excited to know that we have now glass bottom chip in Dynamic 42, and I'm looking forward to use that chip system. Definitely, if we focus on our aim is to establish a system that mimics intestine morphologically and functionally, and to analyze the epithelial cell regeneration after infection and treatment in organoids and on chip. So with chip system, we could see similar effect as we can see it in vivo studies. Organ on chip system was the best system that I could ever imagine to fulfill my aim.